Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the second webinar in the Women's Health 2024 series. Today's webinar is titled Metabolic Mechanisms that Contribute to Pregnancy-Induced Cardiac Growth and Its Reversal, featuring Dr. Helen Collins, Assistant Professor of Medicine at the University of Louisville. Today, she'll give us an overview on cardiovascular changes in healthy pregnancies, risks for cardiovascular disease during pregnancy, and her lab's research on metabolic mechanisms behind pregnancy-induced cardiac growth using multi-omics techniques. Before we get started, we would just like to acknowledge our partners for the series, the American Physiological Society, the Society for Women's Health Research, and Scientist.com. Specifically, we'd like to give our thanks to today's host society, the American Physiological Society, for partnering with us on Dr. Collins' event. We'd also like to say thank you and give a shout out to all of our series sponsors. None of this would be possible without them. I'm Sydney Otamanchuk from the events team at Scientist.com, and I'm happy to be your host for today's event. Before I welcome Dr. Collins to the floor, I'd like to cover a few housekeeping notes to help you all get the most out of your experience today. First, have a look at the interactivity panel at the right of your screen with 4 tabs: chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A and polls during the webinar, as well as access all webinar resources. We want this to be an interactive experience for you, so please send in questions, thoughts, and comments using the Q&A tab. We will try to get to as many of your questions as we can at the end of the webinar today. When a question is added into the Q&A panel, you can upvote the question to increase its position on my list by clicking a thumbs up button underneath it. This is always helpful so I know which questions to address first. You can also explore some links and documents in the handouts tab to learn more. Lastly, if you're having any technical issues, please try refreshing your browser and ensure you're using Google Chrome to access this event, as it is the most stable browser. If you're still having any trouble after you refresh, you can use the private chat function to communicate with me by sending a chat in to Sydney, and I'll help you to troubleshoot. So, Dr. Collins, I will bring you on now to join me. Good morning. Thanks for being here. With that, I'll let you take it away with your presentation.
Okay. I'd like to thank the American Physiological Society, the Society for Women's Health Research, and Inside Scientific for the invitation this morning to present in the 2024 Women's Health Webinar Series and to discuss ongoing and completed studies in the lab focused on pregnancy-induced cardiac growth and its reversal. So these are the disclosures that I have. And just for those of you that are not familiar with my lab, the mission of my lab is to understand how the female heart adapts in the setting of both physiological and pathological stress. So one of those physiological stresses is pregnancy. And what is important to note, and a key take-home message from the talk today, is that being pregnant in the United States is dangerous. And these data here are the maternal mortality rates in the year 2018. And what you'll note is the US clearly leads the pack here with 17.4 deaths per 100,000 live births. And this is significantly higher than the rest of the developed world. If we now look at the most recent statistics, from 2021, you'll note that this maternal mortality rate has significantly increased from the 2018 statistics of 17.4 deaths per 100,000 live births up to, in 2021, 32.9 deaths per 100,000 live births. So this is a significant increase in a very short period of time, and it also underscores our need in the US in particular to address the crisis that we're facing with respect to maternal mortality. So if we take this to a state-by-state basis, these are at the present time outdated statistics, but you'll note that the maternal mortality ratios between 2005 and 2014, you'll note that from this map here that many of the states in America have denoted by this pink color, and this is associated, the intensity of this pink color is associated with the highest rates of maternal mortality. More recently, this has been updated, and now we have the data for each state from, or most of the states, from 2018 to 2022. And you'll note that if you look at these intensities, now the colors change to blue, but if you look at the intensities here, they've increased. If you look back here, the lower half was 10 deaths per 100,000 live births, up until around 30. But if we look now between 2018 and 2022, we're exceeding 30 deaths, and actually closer to 40 deaths per 100,000 live births. And these deaths are predominantly concentrated within the southernmost United States. And in fact, the states of Arkansas, Mississippi, Alabama, Louisiana, Tennessee, and Kentucky make up some of the highest maternal mortality rates in the US. And this is also associated with the highest degree of maternal health care deserts. So this suggests to us that we need to also, on a state-by-state basis, do more to address maternal mortality. So if we focus now on Kentucky, which is the state that I'm currently located in, and we look at some of the deaths that have occurred in Kentucky, we'll note, if we look back to the 2018 statistics from the first slide, we'll note that we saw 17.4 deaths per 100,000 live births. At the very same time in Kentucky, the average was significantly higher than this national average. An abysmal rate of 140.9 deaths per 100,000 live births. So this is significantly higher than the national average, and it's also the case in several of the other southernmost states in America. And what's most alarming is that in many of the cases in this maternal mortality review, it was noted that many of these maternal deaths were preventable. And in particular, in Kentucky, around 90% of these deaths were deemed preventable. So we have to really do our utmost to address these issues more so. What is also important to note with respect to maternal mortality is that maternal mortality rates are highest in Black women. You'll note here from this most recent data between 2018 and 2020 that in comparison to non-Hispanic white and Hispanic women, that non-Hispanic Black women had significantly higher rates of maternal mortality, and it's steadily increased year upon year. So this suggests to us that we have to do more to address the racial disparities that exist within maternal mortality. If we also know that some of the states with the highest maternal mortality rates also had the highest share of Black births. So this also tells us that we also need to significantly address the issues that we're facing with the racial disparities that exist within the maternal mortality, to have a not only a better understanding of what's driving those increased rates, but also to try to address them in a meaningful way. So we see that maternal mortality rates in the US are significantly increased. And why might that be? Well, cardiovascular disease is the leading cause of maternal mortality. And actually, in the US, cardiovascular diseases comprise around 15% of all pregnancy-related deaths in the US. And so what does this cardiovascular disease comprise of? Well, it comprises of several different cardiovascular diseases which can arise during pregnancy, such as arrhythmia, cardiomyopathy, valvular disease, and other diseases such as those which arise during pregnancy, such as peripartum cardiomyopathy and other gestational related diseases that can impact cardiovascular function, and adaptations such as gestational hypertension, preeclampsia, and gestational diabetes. So what's important to note is that pregnancy is associated with cardiovascular clinical symptoms, and these are normal symptoms that occur during pregnancy, which are the result of the adaptations that we do see with maternal systemic and cardiovascular physiology. That are those adaptations which occur are necessary to facilitate the maternal fetal circulatory demands, but they're also associated with clinical symptoms, for example, such as increases in heart rate, also breathlessness upon exertion. But what's important to note is that the exacerbation or the extreme presentation of some of these symptoms is associated with or is suggestive of cardiovascular pathology. So it's important to be aware of the symptoms or the exacerbation of those normal symptoms to then be in a well-educated position to seek out medical attention. So, for example, if breathlessness upon exertion goes to extreme breathlessness upon rest, that's an indicator of some underlying cardiovascular pathology, such as also when we see resting tachycardia going to just extremes of tachycardia or arrhythmias. It's appropriate that we educate women on the knowledge of these symptoms so that they can seek appropriate medical attention, and that could also help reduce maternal mortality if women are more educated on what to look out for. So, as I mentioned, there are several risk factors that can drive maternal cardiovascular disease. Over the last decade, advanced maternal age has arisen as a significant risk factor. You'll note here from this the recent statistics from 2021 and between 2018 and 2021, that women that are 40 years of age and over have significantly increased maternal mortality rates in comparison to younger women. So that is a significant risk factor for maternal cardiovascular disease. Also, multiple gestations and multiple pregnancies are a risk factor. Also, I am part of the Division of Environmental Medicine, so it's important to note that that smoking and vaping and exposure to environmental toxicants and pollutants can also be a contributing factor to maternal cardiovascular disease. Also, several metabolic related factors such as poor nutrition, obesity, diabetes, hyperlipidemia, sleep disordered breathing are thought to contribute to maternal cardiovascular disease. And then also several pregnancy and women's health related factors contributing also, such as gestational hypertension, preeclampsia, diabetes, preterm birth, and then also lactation, which is more important in the setting of peripartum cardiomyopathy, where inappropriate cleavage of the lactogenic hormone prolactin is thought to have anti-angiogenic effects and contribute to maternal cardiovascular disease. So knowing the risk factors and the maternal mortality rates, what we need to do is increase awareness amongst healthcare providers, amongst women, amongst researchers, and the general public about the maternal cardiovascular disease rates and maternal mortality rates, so that we can begin to tackle this in additional detail. And then also improve access to healthcare. As I mentioned, some of the states that are most afflicted by the highest maternal mortality rates are also those which are associated with increased maternal healthcare deserts. So we have to improve access to healthcare and then improve existing pre and postnatal healthcare Usually within women, postnatal health care ends around 6 weeks postpartum. However, it's been noted that several cardiovascular diseases which arise during pregnancy also extend well into the postpartum period, up to at least 1 year. So it's important for us to tackle that, what's been termed as the fourth trimester. And then also we need to consider chronic health conditions, because women with underlying health conditions, some of these can become exacerbated or present during pregnancy. So we need to manage those appropriately. And then also tackle the racial disparities that we see within maternal mortality. And then ultimately, if we have early detection and timely intervention, we can, in fact, help reduce the maternal mortality rates, because if we had a biomarker or something that we could look at in at-risk populations, we could also intervene in a timely manner. So it's important for us to be aware of these factors and to consider maternal cardiovascular disease, because it's recently been shown that maternal cardiovascular disease has a long-lasting impact on female cardiovascular health. This recent study in the Journal of the American Heart Association took women under the age of 25, between 25 and 35 years plus, and irrespective of age, they found that a presence of a cardiovascular complication during pregnancy increased both all-cause mortality and cardiovascular mortality. So not only does this show us that maternal cardiovascular disease has a long-lasting impact on female cardiovascular health, but it shows us that we need to do more to tackle maternal cardiovascular disease. But in order for us to first do that, we need to have an understanding of the normal cardiovascular adaptations and changes that occur during a normal, healthy pregnancy. So, what are those changes? So, it's recently been shown that there are several systemic and cardiovascular adaptations that occur during pregnancy, and these are changes which occur in several different species and in women. And I'll highlight a few of those cardiovascular adaptations. We see an increase in cardiac output. We see changes in blood volume. And we also see changes in vascular compliance and peripheral resistance. But what's most important to this talk, we see there are changes in systemic and cardiovascular metabolism, but also the development of pregnancy-associated cardiac growth. So it's been long established that there's a physiological cardiac growth that can occur with pregnancy. And this is different from what you would typically see with pathological cardiac growth, because with physiological cardiac growth, such as that seen during pregnancy, many of the underlying factors are different. For example, with pregnancy, function is maintained, it's thought to be reversible, and this is unlike what you see during pathological cardiac remodeling. So there are also several proposed mechanisms that are thought to contribute to pregnancy-induced cardiac growth. So there are several hormonal changes that occur during different trimesters during pregnancy, such as changes in estrogen, progesterone, prolactin, and these are thought to have specific effects on the cardiovascular system and also systemic physiology during pregnancy. Also, the mechanical stress imposed on the heart, such as the release, can result on the release of growth factors and release of cytokines, which could also contribute. Also, the increased cardiac workload placed on the heart through the volume overload that we see, and the need to increase cardiac output to meet the maternal and fetal circulatory demands. And then there are other factors that have been considered, such as nutritional factors. But despite these proposed mechanisms, the actual specific molecular mechanisms that contribute to pregnancy-induced cardiac growth are not well understood. So that was a key factor in the studies in my lab. It was to try to decipher some of these molecular mechanisms to understand pregnancy-induced cardiac growth more so. So in the lab, we have studied FEB-NJ mice, and for the most part, we've utilized 4 groups: a non-pregnant diestrous group, a mid-pregnancy group, which reflects day 8 of pregnancy within this strain, a late pregnancy group, which reflects day 16 of pregnancy, and then a 1-week postpartum group, where the dams are maintained with their litter and allowed to essentially rear this litter and lactate. And what we've seen in our studies is that during pregnancy, in addition to the increases in body weight that we observe during pregnancy, we also see increases in heart weight to tibia length at all stages of pregnancy and post-birth. We also see, at least at the post-birth time point, the indications of concentric hypertrophy as seen through an increase in cardiomyocyte cross-sectional area. And we also do not observe any changes in overt cardiac function as assessed via ejection fraction, but we do see characteristic increases in left ventricular end diastolic and end systolic volumes, and then also increases in cardiac output mediated in our hands through an increase in stroke volume. And then also in line with the criteria that I previously showed that define physiological hypertrophy, we also don't see any indications of fibrosis or any activation of the fetal gene program. Also, we have looked at this in the commonly used strain C57 Black 6 mice in comparison to FEB mice, and we also see, irrespective of strain, we see this cardiac growth response during pregnancy. So having established this pregnancy-induced cardiac growth in the lab, we wanted to understand the molecular mechanisms that govern this pregnancy-induced cardiac growth. So it's well established that metabolic changes can contribute to cardiac remodeling. This has been well established more so in the realm of pathological remodeling, but it's clear that during pregnancy, there are several changes in circulating substrates that can be utilized, but also there are changes in cardiac metabolism. And what was supportive of at least the contribution of metabolic changes to pregnancy-induced cardiac growth was studies by the Rainey lab, in which they found that that PDK4 or pyruvate dehydrogenase kinase 4, which inhibits pyruvate dehydrogenase, was significantly increased in late pregnant hearts, and this was associated with this was primarily driven, through an increase in the pregnancy hormone progesterone. But what was most exciting about this particular study was that when they inhibited PDK4 with dichloroacetate, this inhibited cardiac growth during pregnancy, which alluded to the fact that potentially reduction in glucose oxidation was important for cardiac growth. So in order to understand the molecular mechanisms driving pregnancy-induced cardiac growth, we took a multi-omics approach to understand this. And what we have done is bulk RNA-seq of the maternal heart using our 4 groups. And essentially what we find, this is a heat map here of the top 50 transcripts, and with all the heat maps in the talk, red indicates high abundance and blue indicates low abundance. But what we see was with mid-pregnancy, this is associated with increases in transcripts associated with vascular endothelial cells, angiogenesis, cell proliferation. If we move to post-birth for a second, this is associated with increases in transcripts associated with the extracellular matrix and fibroblasts. But if we move back to the late pregnancy time point, this is associated primarily with increases in transcripts associated with metabolism. And also, we were successfully able to corroborate the Irani lab study finding, in which we also were able to show an increase in PDK4 expression in the late pregnant heart. In order to validate this further, we performed some immunoblot studies, and we were able to show that PDK4 at the protein level was increased in the maternal heart at the late pregnancy time point. And we also found that at late pregnancy, there was a reduction in the phosphorylation and thereby activity of PFK2. With our transcriptomics data set, we did note that the ketone body enzyme beta-hydroxybutyrate dehydrogenase 1, or BDH1, was significantly increased during pregnancy. So we also validated that change at the protein level and found that BDH1 was in fact increased in the maternal heart at all stages of pregnancy and post-birth. So what we think is happening is that we're observing a reduction in glucose catabolism at both the level of PFK and PDH, and this is driving glucose into these pathways here, which are ancillary biosynthetic pathways, and these pathways will give rise to the building blocks that are necessary to facilitate cardiac growth. And whilst this is occurring and whilst glucose is being diverted to these pathways, what we believe is happening is that ketone bodies acting via BDH1 are maintaining metabolism. So to begin to understand this more so, we next decided to perform bulk metabolomics to assess whether we saw any increases in metabolites associated with these ancillary biosynthetic pathways. So this is another heat map of the metabolite changes in the maternal heart. And what you'll appreciate is in going from non-pregnant to mid-pregnant, there's very little change in the metabolite profile there, with the bulk of the metabolite changes occurring both during late pregnancy and post-birth during heightened periods of cardiac growth. If we look at the late pregnancy time point, this is associated with increases in amino acid metabolites, specifically those of the urea cycle, where we see actually homoarginine is significantly increased in the late pregnant heart. In addition, we see several other amino acid metabolites increased during late pregnancy. In addition, we see increases in fatty acid metabolites. For example, we see several sphingomyelins and long-chain fatty acids metabolites increasing during that time point. If we move across to the post-birth time point, we still see the amino acid metabolites from both the urea cycle, but now we see increases in polyamine metabolites. And this is of particular interest because polyamine metabolites have been indicated in hypertrophy, but they've been indicated to contribute to pathological hypertrophy. So it's area of investigation that we would like to look into to see their contribution to physiological remodeling. In addition, we see increases in nucleotide metabolites and glycerophospholipid metabolites. These three pathways are increases in metabolites in these three pathways are indicative of glucose being diverted to those ancillary biosynthetic pathways of glucose metabolism to facilitate the production of building blocks necessary for cardiac growth. However, bulk metabolomics only gives us a snapshot of what's happening at any given one time. So we wanted to now determine whether there was, in fact, increased incorporation of glucose into these specific pathways. So in order to do this, we utilized our 4 groups and we provided them with a liquid diet containing C13 labeled glucose. And this diet was provided for 18 hours, which we've shown is sufficient to label the ancillary biosynthetic pathways of glucose metabolism. After which the hearts are freeze clamped, and then the stable isotope resolved metabolomics and deep network tracing is performed. So as I'll show some changes here, but it's important to note that these are just representative changes. There are a large abundance of changes that do occur, but for paucity within this talk, I'll only discuss a few examples. So essentially what we do find is there's increased incorporation of glucose-derived carbon into these ancillary biosynthetic pathways of glucose metabolism. We see increased incorporation into nucleotide production as evidenced by this increase in UMP. We see increased incorporation of glucose into glycophospholipid pathway, such as this increase in glycophosphoglycerol that we observe. And then increase incorporation into amino acid metabolism, such as the changes that we observe here with L-methionine. But what's important to note is that given the lack of knowledge in this area and some results that we have, we included an additional group, which is this post-birth non-lactation group. And I'll tell you why that is so now. So what we found in the lab is that lactation has a profound effect on pregnancy-induced cardiac growth. So we've included our 4 groups that we've utilized during these studies. But for the purposes of this, we've included this post-birth non-lactation group. And it's matched with this post-birth group. However, the only difference with this group is immediately after birth, we remove the pups to reduce lactation. And when we do this, we find that body weight reverses in the post-birth non-lactation group to a comparable level such as that seen during mid-pregnancy. And then also we see with respect to the heart size that hearts reverse to a size comparable with late pregnancy. And this is just 1 week postpartum with both of these groups. So lactation is having a significant effect on cardiac growth. So because of this, we included this particular group in our stable isotope resolved metabolomics studies. And what we essentially find is a reversal of many of the changes that we observe with respect to the incorporation of glucose into the ancillary biosynthetic pathways, such that there are significant reductions in the UMP enrichment, the glycophosphoglycerol enrichment, and L-methionine enrichment. So this tells us that lactation is having a profound effect on pregnancy-induced cardiac growth and is impacting glucose metabolism in the heart. So I mentioned at the start of the talk, we took a tri-omics approach to investigate mechanisms contributing to pregnancy-induced cardiac growth. And so we also performed cardiac proteomics of the maternal heart. And so this is a heat map of our cardiac proteins. And you'll note once again, similar to what we had seen with our metabolite profile, that there are few changes that occur at the protein level between non-pregnant and mid-pregnant hearts, with the bulk of the changes we observe occurring in late pregnancy and post-birth when we see that heightened cardiac growth. And specifically, late pregnancy and post-birth are associated with significantly lower expression of mitochondrial complex subunit proteins, specifically cytochrome C oxidase and NADH ubiquinone oxidoreductase subunits, several histones, and several contractile proteins. So to begin to understand the physiological consequence of this reduction in mitochondrial subunit complex proteins, and given our interest in the maintenance of mitochondrial metabolism during the redirection of glucose into our biosynthetic pathways, we next examined mitochondrial function within the maternal heart using isolated mitochondria. But before we began that, we also reanalyzed both our transcriptomics data set and proteomics data set and aligned them along with the mouse mitocarta to have a greater understanding of the mitochondrial transcriptional and proteomic changes that were occurring, and to also reincorporate some of the mitochondrial transcripts that are often excluded from RNA-seq analyses. So when we did this, we were able to show many of the changes that we find at the whole heart level, such that we see an increase in BDH1 transcript at late pregnancy. We also see it maintained at post-birth, but we also see several additional mitochondrial transcripts changing within the heart during various stages. And we also see switching of some of the transcript expression levels in going from late pregnancy to post-birth. When we look at the reanalyzed proteomic changes, we know that many of the proteins that are changed are those associated with the mitochondrial electron transport chain. And we are getting indications from some of these changes that there are several potential changes in the complexes, the subunits during each stage of pregnancy. So that would be something that we would investigate further in future to determine whether there's specific metabolic channeling during specific pregnancy stages. So as I mentioned, we wanted to then determine mitochondrial functional changes. So in order to do this, we took our 4 groups. Unfortunately, we were unable to include the post-birth non-lactation group in this due to a technical issue, but we're actively performing new studies now in which we will be including that group. But with our 4 groups, we isolated cardiac mitochondria, and their primary fate was to undergo respirometry using a Seahorse XF extracellular flux analyzer in response to several substrates, such as a carbohydrate-derived source such as pyruvate malate, fatty acid source octanylcarnitine malate, also an amino acid-derived source such as glutamate malate. And then with the remaining mitochondria that were left over, we saved them for immunoblot analysis to interrogate key mitochondrial protein changes. So with every study that we do, we also reconfirm the hypertrophic response. So we were also, even though this was a lower biological N, we were able to show that we see our characteristic changes in body weight during pregnancy, and we also see our increase in heart weight to tibia length during all stages of pregnancy and post-birth. Given some of the reanalyzed transcriptomics data indicated that several mitochondrial dynamics proteins could be changed in expression, this didn't really result in any change in mitochondrial yield between any stage of pregnancy and post-birth. So we next perform the respirometry, and what we find is when we give a pyruvate malate mixture, which is a carbohydrate derived source, and a amino acid derived source such as a glutamate malate, we see that there are primarily postpartum or post-birth mitochondria are able to avidly utilize both of these substrates, and this is highly efficient as determined by this state 3 to state 4 respiration, also known as the respiratory coupling ratio. We also did not observe any changes with the fatty acid derived source or also succinate rotenone between each group. So this shows us that post-birth mitochondria are able to avidly utilize these 2 substrates as an efficient energy source. So we next wondered the implications of potentially giving these isolated mitochondria a ketone body derived source, given the changes that we observed in BDH1. And also, this is because in the literature, changes in ketone body metabolism have been thought to contribute to metabolic remodeling in the heart. Many of these studies have been performed in the context of pathology, but they indicate that the BDH1, which is the primary ketone body enzyme, and also ketone bodies could be a metabolic stress defense, and that could also contribute to physiological remodeling. And what's important to note is ketone body metabolism does change during pregnancy. Pregnancy is associated with increases in circulating levels of both the primary and secondary ketone body, primary one being 3-hydroxybutyrate or beta-hydroxybutyrate, and then the secondary one being acetoacetate. But essentially, BDH1 is not only important in the generation of beta-hydroxybutyrate, but also its use in extrahepatic organs. However, the role of ketone body metabolism, especially in the heart, is not well established. So we decided to give our isolated mitochondria 3-hydroxybutyrate to see if they would utilize that as an energy source. And so essentially what we find is when we give isolated mitochondria 3-hydroxybutyrate, we find that post-birth mitochondria are able to avidly utilize 3-hydroxybutyrate. However, in comparison to both the carbohydrate and amino acid derived sources, the utilization of ketone bodies is less efficient. In order to then establish whether this is due to any changes in mitochondrial expression of key enzymes, we confirmed BDH1 expression in isolated mitochondria and found increases in BDH1 expression in the postpartum heart in line with these findings here. And also in line with these findings, we see a significant reduction in PDK4 expression. So in order to understand the role of BDH1 in the heart during pregnancy, we have recently generated a tamoxifen-inducible cardiomyocyte-specific BDH1 knockout mouse. And our preliminary studies suggest that these mice have normal growth responses and comparable growth responses to wild-type glutamate controls during pregnancy. However, the ketone body response, the circulating ketone body levels are slightly reduced in the knockout mice. And then also what we find is the hypertrophic response that we observe in wild type mice is not necessarily seen in the knockout mice. So these studies are actively ongoing now, and we'll be happy to present those findings in more detail in the future. So hopefully what I've shown today is that during pregnancy, the heart increases in size. And this is associated with increases in transcripts associated with the cell cycle, angiogenesis, cell proliferation, and the vascular endothelial cell compartment. We also see that during late pregnancy, the heart increases further in size, and this is associated with increases in transcripts associated with metabolism. And then we also see signatures associated with a reduction in glucose catabolism and an increase in BDH1 expression. And then when we get to post-birth, we see changes in transcripts associated with extracellular matrix. We see beginnings of the reversal of some of the metabolic changes in that we see a reduction in PDK4, but we also see sustained BDH1 expression. And we also see at this time point that mitochondria are able to avidly respire using ketone bodies. And between these two time points associated with heightened cardiac growth, we see the increased abundance of pro-growth metabolites, such as amino acid metabolites, glycosphosphorlipid, and nucleotide. So hopefully what I've shown is that pregnancy-induced cardiac growth involves coordination of several metabolic pathways, but with like any body of research, we have many unanswered questions that remain, such as the contribution of liver-derived metabolites to pregnancy-induced cardiac remodeling and whether they play a role in pathology. Ketone bodies have been shown to be protective. They're generated in the liver. We also see that the metabolite that's most abundant in the late pregnant heart is homoarginine, which is also produced in the liver. So we need to know and establish the role that they might play. Also, the extent to which other cell compartments play a role is evident from our transcriptomics data, that there are coordinated changes in gene expression that occur, which are likely to reflect coordinated changes in physiological remodeling with those particular cell populations. So we have to do more studies there to interrogate that. And then we need to address the impact of circulating hormones, the placenta, and maternal fetal interactions and lactation on this remodeling response. Also, we need to determine the nature of the hypertrophic response that we see and the impact of successive pregnancies on pregnancy-induced cardiac growth. I do have a little piece of data for the last 2 bullet points here. So with regard to the nature of the hypertrophic response, what we did see earlier in the presentation is that despite the heart gravimetric size increase, that didn't correlate with a change, at least at late pregnancy, in cardiomyocyte cross-sectional area. Although this is most likely to be the result of eccentric remodeling at that time point, it could be that there are changes in volume of the cardiomyocyte. However, we've been able to rule this out by doing some analyses of the wet heart weight to tibia length and the dry heart weight to tibia length, where we see those changes are maintained when we've dried out the heart. And the fluid content in the heart is comparable between all time points there. So this leads us to indicate that we need to do additional studies to determine myocyte length. Also, the impact of successive pregnancies. Most of our studies have been just the result of one pregnancy, but we've recently looked at the impact of up to 4. And what we see is that with each subsequent pregnancy, the heart increases in comparison to non-pregnant controls, but it doesn't increase any more so between the number of pregnancies in the normal healthy condition. And this is associated with increases in myocyte cross-sectional area. So this is something that we're actively interrogating in the lab now to understand further. So with that, I'd like to thank my faculty mentors in the center, people in the lab that have assisted with these studies, specifically Emily, Kyle, Richa, Avery, and also other members of the center that have helped obtain some of the results here. And then our external collaborators and R01 grant consultants. And then I'd like to thank all the funding bodies that have made this research possible. And then lastly, I'd like to thank you all for listening, and I'll be happy to take questions now. But if you would like to reach out to me directly, here are my contact details. And here is a website address for our center if you'd like to see what cool science we're all doing in Louisville.
Wonderful. Thank you so much, Dr. Collins. That was a really great presentation. I'll leave your slide up just for another moment in case people need to write that down. And I'm going to just give you a second to catch your breath here before we jump into the Q&A. So I'll see you back in a moment. In the meantime, I did want to just remind the audience to please submit your questions into the Q&A tab. We will try to get to as many as we can live in the last about 10 minutes that we have left of the webinar. And then if you do have to leave early, I just would like to leave a pop-up link for you to complete our post-event survey. You can let us know your thoughts on your experience today and the webinar. You can also send in some webinar topics you'd like to see in the future, and also submit any questions for Dr. Collins in that survey as well. So we do really appreciate your feedback and taking the time to do that. Okay. So switching back here, we have started to get a bunch of questions rolling in. Looks great. So Helen, I'll bring you back on now. Okay. Thanks again for that presentation. It really was great. The first question for you, in your tamoxifen-inducible cardiomyocyte-specific knockout model, what did you use? Was it tamoxifen-inducible Cre recombinase? And if so, technically, how did you do it? Does it affect estrogen signaling and activity?
So we use the commercially available um, tamoxifen-inducible Cre produced by Dr. Malkington. And so, in order to do those studies, we provided tamoxifen chow to our mice. This is different to the standard procedure that many investigators do with the IP injections of tamoxifen, which occur between, I think, some people do it as short as 2 days, some people do it up to a week. But we opted for the chow method because this produced a reliable knockdown of BDH1 in the heart. And this was given for 3 weeks with at least 2 weeks washout. And what we had to do a, such a washout like this because actually um, tamoxifen does impact estrogen signaling. So we have to make sure that it has left the system before we start to perform timed pregnancy studies, because we don't want to go into a study there where we're adversely impacting estrogen, which is required, especially most importantly during earlier pregnancy, as one of the key hormones that increases earlier. Um, what was the second part of the question?
Yeah, they did submit actually a clarification that they wanted to know specifically how you induced pregnancy, whether it was prior to or
Oh, okay.
after deleting the genes, and how early was the tamoxifen injection. Hopefully that helps.
So just a clarification, there was no injection. It was a chow. And we had after 3 weeks of chow provision, we switched back to a normal standard chow for at least 2 weeks before we got the mice pregnant. Um,
Okay,
yeah,
perfect.
so.
Thank you. Um, so that we have another question here. The mouse hearts seem to recover pretty well after multiple pregnancies. What is the period length between pregnancies? And they also say thank you for the awesome presentation.
Oh, well, thank you very much for attending. And those particular studies, so I I kind of maybe glossed over that a tiny bit, but so we did a full pregnancy and then a full lactation period. So what we did is obviously for one pregnancy it's that, but between uh, two pregnancies, so we had two pregnancy lactation cycles. This was at least 3 weeks between starting again, um, because we have also got data to suggest that at least within 3 weeks after birth, there is um, a regression of the heart size to almost what we would see during mid-pregnancy. We unfortunately, in our hands, have not seen it ever go back to comparable levels that are seen during non-pregnancy, even up to a year. So,
Okay,
um,
interesting. All right, thank you for sharing the information. Um, can you share what the cardiac biomarkers you should screen for during pregnancy?
oh, well, I know there are some that are already candidates out there, like pro BNP and troponins and such. Um, I have done some studies where I've looked at the circulating proteome, and there are some additional changes. Also with I think peripartum cardiomyopathy, that people often will look at the circulating S-FLT1 levels as well. But I think problem that is faced as well is that several of these factors do increase normally. So caution's probably needed with assessing their levels, and it's probably more so that it's a concentration dependent effect where it reaches a certain level during a normal pregnancy, but it's heightened during a pathological associated pregnancies. But then we would have to take care of um, those levels more so. And then obviously there's person to person variation. There might not be a one fits all biomarker. It would likely have to be a panel of biomarkers that we would have to look at. But that's what we hope to, at least in our preclinical studies moving forward, at least come up with some circulating biomarkers that are associated with several different pregnancy-induced cardiovascular disease states.
Okay. Yeah, makes sense. Thank you. I think we have time to squeeze in this very last question. So bear with me to read it out. It seems it would be useful to compare healthy maternal cardiac growth to pathological maternal cardiac growth. Do you have any plans to do this with your animal model, perhaps with an exercise or dietary treatment prior to pregnancy to predispose the animals to healthy versus unhealthy progression during pregnancy?
Uh, I guess if you mean like the earlier studies without the BDH1 knockout and just adding a comorbidity on top of that, such as like adding a high fat diet to induce obesity or such. That's something that we would like to do, and we have started to interrogate at least the impact of high fat, high sucrose diets with varying salt contents as well, to examine the impact of that. We haven't yet combined it with like the BDH1 studies yet because they're still ongoing. But and they have significant phenotype that um, is um, tricky to manage. But with respect to those studies, we will do them in the future, and the ones that we have started indicate that, at least actually in our hands, the low fat diets that we give are actually seem to cause more dilatation than the high fat, high sucrose diets, which actually caused what I would term like positive changes in function. But that's something that we need to keep a track of moving forward. And then because we also wanted to have a firm foundation of what occurs during a normal pregnancy before we added on extra biological variables.
Right. Okay, I think that all makes sense. Thank you so much for answering those questions. We have reached top of the hour. So again, I want to thank you for being here, for your time and expertise. It was such a pleasure to have you with us, and I hope you enjoyed yourself.
I did. Thank you very much again for the invitation.
Of course, you're very welcome. All right. And thank you so much to our live audience. We know how busy you can be, so we really appreciate you being here with us. And I hope that you also enjoyed your experience today. Please join us for the next event in the series, which is on Wednesday, October 30th at 11 a.m. Eastern Time. You will receive a reminder email soon with the link to log in. Make sure you block it off in your calendar so you don't miss it. A quick reminder, a recording of today's webinar will be available soon, so keep an eye out for an email that will give you access to that. Additionally, that survey will appear at the end of the webinar. We do read all of your comments and really appreciate your input on your experience today. And as I mentioned before, you can let us know what webinar topics you'd like to see in the future. Finally, if you still have questions for our speaker, you can submit them in the survey and we'll be sure to pass them along to Helen. In closing, we hope you enjoyed this Inside Scientific webinar. Another quick thank you to our partners and sponsors for helping to make all of this possible, and I hope you all have a great rest of your day.